Hello and welcome to another BYU-181 video. Today we'll be talking about the genetic code and the central dogma of DNA, and that is how we go from DNA to protein via RNA. So DNA to messenger RNA to protein. This path is quite complex, so that's what we'll be discussing. As mentioned in previous videos, and I'm sure you're aware that you probably learned this in Bio 180, but if here's our cell, there's a nucleus. The nucleus has got DNA inside it, and that DNA is transcribed, a portion, not all of it, but say this little segment here, we transcribed into RNA. And that RNA, this little segment of RNA, comes out into the cytoplasm, where ribosome protein RNA machinery finds this little messenger RNA right here and translates it, and as it translates this, it produces a polypeptide chain, or this is our protein. So we went from DNA to RNA right here. The RNA went out to the cytoplasm, was translated into a protein. So into a protein. DNA to RNA, messenger RNA, is done by transcription. And then RNA to protein, how it's converted, or how the system, the ribosome, knows how to read an RNA and make an amino acid chain. The process is called translation. So keep that in mind. We'll discuss that more as we progress throughout this course, but for right now we're going to talk about the DNA code, how we go from RNA to protein. So as I drew, here's our messenger RNA. The messenger RNA carries the genetic message. On it there are a bunch of nitrogenous bases. Adenine, uracil, guanine, uracil, adenine, guanine, cytosine, you name it. Everything but thymine is going to be present on this single-stranded RNA. Guanine, uracil, blah 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 blah, whatever. So how the machinery, how the ribosome reads it reads it in groups of 3. Instead of reading each individual nitrogenous base, the ribosome reads in groups of 3, and these are called codons. We have another codon here, 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 and so on and so forth. And so as the ribosome's reading, it will read this, recognize the codon, and build the appropriate attach the appropriate amino acid to the chain. And each one, this codon might code for one amino acid, this codon might code for the same amino acid. Codons can code for the same amino acid, but maybe this codon codes for a different amino acid, this codon codes for a different amino acid, and they'll be linked together by the ribosome, and this will be our polypeptide chain, which eventually becomes, as it folds into its quaternary tertiary structure, whatever, it becomes a protein. Now, some terms you need to understand is that the genetic code is unambiguous. I probably butchered this spelling. Unambiguous, this word here. What does it mean? It means that one codon codes for one amino acid. One codon codes for one amino acid, that's important. One codon cannot code for 2 different amino acids. and then the other term you need to know is that the genetic code is degenerate. This is important and means that multiple codons can code for the same amino acid. Amino acid here. So again, the genetic code is unambiguous. So if the ribosome reads a codon, that's going to translate into one specific amino acid. However, it's degenerate, meaning that there are multiple codons for the same amino acid. So you could have GUU, that codes for, I'm going to make it up, it codes for amino acid 1 only, but GUA also codes for the same amino acid 1. I don't even know if this is true. The genetic code is, you don't have to memorize it, you don't need to know it. There are tables, there's a wonderful table that's got all the possible amino acids, and codon combinations possible, but you don't need to memorize it, and this example is probably not even true. Whether these two codons, GUU and GUA, code for the same amino acid, I don't know, but it's not important, it's just for the example. And you won't have to memorize it, like I said. If any problems like this to do with figuring out what something would translate into, any problems like that on a test, you would just be given the genetic code. So it's nice and easy. Now, how is that possible, that the code can be degenerate? How can multiple codons code for the same amino acid? Well, we have 64 codons but only 20 amino acids. If that's the case, with more codons than amino acids, there must be multiple codons that code for the same one. And how we know there are 20 amino acids. They've been listed, they have different characteristics of polarity and acidity, basicity, and so forth. You don't need to know too much about them specifically, but how we know there are 64 codons? Well, if we have the combination of nitrogenous bases as adenine, uracil, guanine, cytosine, that's it for RNA, and DNA would be a T instead of a U, but we've got these 4 options, and they're grouped into groups of 3. So there are 4 possible options. We have 4 options for the first amino acid, then we have another 4 options for the second amino acid, and another 4 options for the third amino acid. First, second, third, and we just multiply these together. So 4 to the power 3 equals 64 codons. That's how we know that there are 64 codons, and they can all code for different amino acids. And if you go further in genetics, you'll learn why multiple codons code for the same amino acid. It's called the wobble effect. It's pretty cool, and you'll learn about that if you take Bio 375. And that's it. So let's quickly review. The central dogma of the genetic code, the central dogma of DNA is that DNA can be changed into, transcribed into mRNA, and then translated into proteins. This happens within the cell. The transcription happens in the nucleus. Translation happens in the cytoplasm. mRNA molecule contains codons, and the codons are groups of 3 amino acids, and they are read by the ribosome and translated into different amino acids. The genetic code is unambiguous, meaning that one codon can code only for one amino acid, but there are multiple codons that do code for the same amino acid. So I guess a bit of clarification. One codon cannot mistakenly code for a different amino acid. It has to code for one specific amino acid. And then we know that there are 64 codons that code for the 20 amino acids, and that kind of proves the degeneracy of the codons, because 4 to the power 3 is equal to 64. I hope that helped. What you'll have to understand and do more concerning this is understand the Beadle and Tatum experiment and how that kind of proved the idea that DNA is truly responsible for the creation of proteins. They did an experiment where they mutated different genes and looked at the effect on biochemical pathways. And I will be covering this in another video, probably titled Genetic Code Beadle and Tatum Experiment, and it's basically the second half of this video. So thank you for watching. I hope that helped. Good luck. And I'm back. I made a mistake. I just want to clarify something I said earlier. I didn't want to record the whole thing again, so I just thought I'd clarify just a little mistake I made. Here are the 3 codons here. They're made of nitrogenous bases. I accidentally said amino acids, but each one is nitrogenous bases. Groups of 3 nitrogenous bases are called codons. The group of 3 of these is called codons. I accidentally said amino acids, my bad. And this, the example of how this one amino acid can code for one codon can code for one amino acid. A different codon here, this UGU, could potentially also code for the same amino acid, and then a different amino acid, different codon, I keep saying that, different codon could code for different amino acid, and where's the purple has different codon could code for another type of amino acid. So sorry, there's a bit of confusion there, but just remember that 3 nitrogenous bases, 1, 2, 3, group together make a codon, and each codon translates into one specific amino acid, or multiple codons. Here, multiple codons can code for the same amino acid, to do with the unambiguity and degeneracy of the genetic code. Sorry about that, I kind of butchered it earlier, so I thought I'd